So why don't we get started? Our first speaker is Dr. Derek Dickhorn, and we're going to hear about pluripotent stem cells.
All right, good morning, and thank you for inviting me to give this talk on some of our recent discoveries that we've made. And this is actually a new project for us. So I'm going to talk a little bit about what are induced pluripotent stem cells, give an update on the work that we've done, and then I'm going to talk about why we think that these are really important, both as a tool for how we understand disease, but also as a means for therapeutic development. Again, this work stems from some earlier discoveries within the Institute, and this was really a collaboration between the Hussman Institute for Human Genomics and the Baskin-Palmer Institute, where they were able to identify a new RP-causing gene, which is called DHDDS. It's the dihydrodolicyl diphosphate synthase, which I'm saying specifically for Betty because I know she wanted to know what that acronym means. And this was found in a family where they had 3 affected individuals and 1 unaffected individual. And it turns out that the parents were heterozygote for this mutation. And this is a mutation which I'm not going to spend a lot of time talking about it because there's going to be a couple of talks later in the afternoon which are really going to go into a lot of what this gene does and how it's functioning and how it may be involved in RP. But it's involved in the post-translational modification of proteins, which is an important process for how proteins are actually going to become functional. So this was identified within this family, and this was a new novel finding. This was done with Stefan Zuschner, who's in the back there, as well as other members of the team from the Hig and from the Baskin-Palmer. And I'm not going to talk too much about that mutation, but that's just the background for the cells that we were using. What I am going to talk about today is about this concept of induced pluripotent stem cells. And we hear a lot about stem cells, particularly naturally occurring stem cells. And we're going to hear some more about this later in this talk, where we're going to hear about embryonic stem cells, about neural stem cells. Well, induced pluripotent stem cells are different than those types of stem cells. Induced pluripotent stem cells means they're induced first as they're experimentally derived. So they're not naturally occurring stem cells within the body, but they're derived from somatic or differentiated cells, which we then make into stem cells. And I'll go through how this process occurs. They're pluripotent in that they have the capacity, once they're made into stem cells, to be made into a variety of different types of cells. So they can be differentiated into a multitude of different types of cells. And this technology is reasonably new, but what it allows us to do is to make patient-specific stem cells. So that is, we will have stem cells which are made from the individual. So they're going to have the same genetic background as the individual from whom they're derived. And this is an advantage because we're interested in studying what impact specific genetic variations have on a disease process. So this allows us to make very, very specific tools to be able to understand how that disease is contributing to this process. And as we'll go over, hopefully we'll also be able to use these tools to be able to develop therapeutics and begin to think about cell-based therapeutic approaches for the treatment of retinitis pigmentosa. And again, this allows us to study the functionality in the disease process, as well as serving as a platform for drug discovery and cell-based. Now again, unlike embryonic stem cells, as I've mentioned, which are derived from human embryos, and we'll talk about them in the next talk, iPS cells are experimentally derived from adult non-dividing cells, and this is usually done from skin cells, but we've also done it from peripheral blood mononuclear cells, and by providing them with reprogramming factors. So this is the basic flow of how this works. You can take a skin fibroblast from an individual, and then you treat these skin fibroblasts by overexpressing a set of transcription factors which are normally restricted in expression to progenitor and stem cells. When these are overexpressed for a long period of time, they're going to change the transcriptional networks within the cells. That is, they're going to change the identity of the cell, so it's going to lose the skin cell or the fibroblast identity, and it's now going to gain a stem cell identity. So these are overexpressed, and we're using 4 factors, OCT4, KLF4, SOX2, and c-MYC. And then over a period of time selection, these are very rare events, you will get colonies of stem cells that will appear. So when we have these stem cell colonies, well, why is this important even for us to be able to do? Well, unlike the skin fibroblasts that they come from, when you grow skin fibroblasts within cultures, they'll grow for a limited amount of time. Actually, skin fibroblasts are pretty decent. They'll grow for about 2 months before they're going to senesce. iPS cells, on the other hand, or stem cells in general, on the other hand, are self-renewing. That is, they are capable of making more of themselves. So this gives us the ability to make enough material to actually be able to study particular processes. The second feature of these stem cells that's important for our ability to be able to use them as disease models and why we think they're going to be so powerful is, as I already mentioned, they're pluripotent. That is, from these stem cells, you're able to derive a wide variety of different types of differentiated cells. You can do ones that are going to be specific for ocular disorders, photoreceptors, retinal pigmented epithelial cells, retinal ganglion cells, but we can also do this for a variety of other types, and people have used this technology to make all kinds of different types of cells, from liver cells, blood cells, pancreas cells, so it's widely applicable to a large variety of scenarios. So the first thing that we wanted to do is that we got these skin fibroblast samples from the family, from each of the members of the family, and we wanted to induce them to make induce pluripotent stem cells. So now what I'm showing is a couple of representative examples of these cells. We've been able to successfully generate them from each of the individuals in this family. And the first thing that we're going to test for is whether or not these actually are stem cells. So we're going to look at them to see that they are pluripotent. The first way we do this is by looking at these, and you can see these are the stem cell colonies here, and they have a very distinct stem cell morphology, and they look just like ES cells. So if you were to put an ES cell colony right beside it, they would look very similar to that. And they are expressing things that they should be expressing if they're stem cells that are not being expressed in the fibroblasts. One of these is NANOG. We have OCT4 expression. And this is by immunocytochemistry. So we basically take the colonies and we stain them, and they're expressing the right markers. We can also do this by gene expression profiles, and we can show that compared to a control IPS line that we have very well characterized in the lab, that they're expressing all of the factors that we would expect to be expressed in stem cells. So you have KLF4, ZFP42, OCT4, and NANOG. And so you'll notice actually some of these are actually some of the factors that we're using to express. But these are actually being expressed from the endogenous loci. So what happens is, once these begin the process of becoming pluripotent stem cells, and once they're initiated, it's a self-fulfilling prophecy. Once they become stem cells, then they maintain that identity within the cell. So these are actually switching from expression, and that's why we look at these additional markers. NANOG is not one of the factors we use. Some people do use it for reprogramming. We don't use it for reprogramming, so that when NANOG comes up, we're pretty sure that these are stem cells. And they also have functional traits of stem cells. So stem cells have high activity of alkaline phosphatase. And so we can stain these for alkaline phosphatase activity and show that they indeed do have high alkaline phosphatase activity as we would expect. And then we can also look at them functionally. So the first step that we do when we're looking at iPS cells or stem cells and the process of differentiation is we make them into embryoid bodies. And basically embryoid bodies are spherical groups of cells which have the potential still to express genes from all three germ layers. So they're basically still, in many ways, pluripotent, but they're kind of one step removed from stem cells towards differentiating. And we can get beautiful spherical embryoid bodies from these cells that we have. So while we were doing this, we were also testing out ways to differentiate this, because obviously the reason we want to do this is so that we're going to be able to look at what is the impact of these specific genetic variations on cells that are relevant to retinitis pigmentosa, namely photoreceptors and retinal pigmented epithelial cells. So we began working with some of our control cells that we saw in the earlier ones, and we're able to take these, and now we're going sequentially from the iPS cells to retinal progenitor cells, and then to what we consider to be fully differentiated mature photoreceptor cells. The way that this is done is by taking the cells and then treating them through a series of different steps where you treat them with different growth factors, which will progressively bring them to a more differentiated or a more mature state. So we begin with them being stem cells, move them into progenitor cells, and then we sequentially bring them down. Thank you, we can see this a lot better now. Sequentially bring them down to the point where they are going to be functionally mature photoreceptor cells. And again, it's a very long process. Photoreceptor cells are some of the most difficult to work with. It takes about 150 to 180 days in culture. We're trying to come up with ways that we can actually modify and streamline this, because my graduate students despair of ever actually graduating when you have to use half a year just to generate cells. This is just some examples of how we looking at them. So this is a progenitor cell, and we want to see that it's actually expressing the factors we would express in a retinal progenitor cell. And it's doing this. You can still see it has the colony morphology, but it expresses PAC6 and MIDF very nicely. Then we can look down at the end of this, where we're looking at specifically things that are going to be expressed in photoreceptor cells. And now we're looking at conorescin in this case, and you can see the nice conorescence staining. And again, obviously the morphology has completely changed. Now we have these nice elongated cells, as you would expect neuronal cells or neuronal lineage type cells to look like. And again, you can see that there's a lot more staining for the DAPI here than are actually in these cells. That's because they're growing on a monolayer of additional cells. When you do these differentiations, you never get a pure population. You always get a heterogeneous population where you have the underlying structural cells as well. And this is actually necessary for the growth of these cells and the health of the culture. So I'm just going to take a very brief time, because I've already had the time flashed at me, to describe why we think that this is actually an important tool. Now, one, obviously this now gives us the ability to take cells from an individual where we have them very well genetically characterized and be able to now study the effect of that genetic variation on very specific types of cells that are going to be disease specific, even if we don't have access to the primary cells. So we can now generate DHDADS specific photoreceptor cells that we can now begin to understand why DHDDS is functioning, or its impact on the functioning of the photoreceptors. Again, can serve as a source for screening for drugs. Normally how screening for drugs is done is that they take whatever cells they can get, which is usually skin fibroblasts, because that's the primary cells. However, we know that drugs act differently and cells function differently, and the impact of genetic variations is different in different types of cells. And obviously this is very specific for individuals who have photoreceptor deficits. And so we can now take these cells, make them into photoreceptors, and use those photoreceptors as a screening platform, as a very highly specific and sensitive screening platform for new compounds that may be able to reverse any type of deficiency within them. And again, we're thinking about this in the future. This could also be a source of cells for tissue transplantation. Now, of course, the benefit of this is that it's autologous. It's taken from the individual, and it can be put back within that individual. Of course, we would need to go through some gene correction in order to do that. And we're going to hear some nice talks later on about some ideas about how to do this by viral transduction and overexpression of the wild-type versions of this. There's other new technologies out there, molecular scissors, where you can actually change the locus itself by replacing the one that has the mutation with the wild-type locus. And so these are some of the ideas that we're going to be exploring. Of course, this work can't be done without great collaborators and people within the lab. This work was done by my team in the IPS lab, Brooke DeRosa, who's a very talented graduate student, Kinsley Bell, another talented graduate student, Jessica Van Baron, who's an extremely talented technician and manages everything for me. Again, this is supported by Jeff Vance and Margaret Paracut Vance, who have been really supportive of the IPS program. It's a new program, a new way for us to look at the genetics of disease and really model them, particularly for neurological disorders. And our great collaborators at the Baskin Palmer Eye Institute, some of who will speak later on today, Byron Lam, Rong Wen, and Jeffrey Goldberg, who I haven't talked about the work we did with him, but it has some very nice work on retinal ganglion cell differentiation from iPSC. So you can ask him about that later on. Thank you for your attention. Of course, we would not have been able to do this if we hadn't gotten the pilot award for Hope for Vision. So all of this work was derived from the funds that they had provided us for this. Thank you.
It's an excellent talk.
So the work that I'm going to talk about started with a postdoc that came to the lab and wanted to work with stem cells. This is about 8 years ago. So he started culturing stem cells, embryonic stem cells that we have got. And he calls me and says, come and see what I'm getting here. I don't know, but I see the stem cells, the colonies, and I see all these little vesicles around there. So I said, oh, something happened, you contaminated your culture, just start all over. So he started all over. The second time, he gets the same kind of thing. By the third time, I said, well, let's characterize what's that. So that's how we started this work. And this work is on microvesicles, these vesicles that are released by the stem cells, embryonic stem cells. So what are microvesicles? So they are little vesicles, 15 nanometers to 1 micrometer, formed from the endosomal compartment and shed plasma membrane of most cells. And they were thought of being inert cellular debris. But now they are recognized as important mediators of intercellular communication. And microvesicles shedding is a distinct physiological process, with some cells shedding microvesicles with a recognizable polarity. So why study them? Well, they are a novel method to transfer bioactive lipids, proteins, and nucleic acids to cells. So they are like physiological transfection agents. That's what we think. Microvesicles may be important for signaling in stem cell niches, and microvesicles may serve a role as paracrine factors in the eye, providing endogenous stem cells with extrinsic cues that may regulate their proliferation. Now, there is evidence for therapeutic potential of microvesicles. And this has been shown with work in blood, with they promote the expansion of hematopoietic progenitor cells. They have been shown mesenchymal stem cells, microvesicles, prevent apoptosis and enhance functional recovery in acute and chronic kidney injury. And in liver, also, they have shown the effect of microvesicles in the regeneration of liver, and also in lung. So the same kind of things have been seen. So we said, okay, let's study these microvesicles, and we found that they contain mRNA, microRNAs, protein and lipids, and that they can also have transgenes that were expressed in the stem cells that release them. And so microvesicles could serve as vehicles for gene delivery. And I'll show you this with GFP. So we had stem cells that were transfected with GFP, and so they are green. In fact, the whole mouse was green because every single tissue in the mouse was green. But the stem cells were green. And so the microvesicles that they released were all green. So we amplified these microvesicles by RT-PCR, and we could see that the microvesicles have the GFP, the EM cells, the stem. Oh, no. How do I go back there? Okay. So is this same one? No. I wanted the pointer, but all the same. You can see here the ESMs. We call them embryonic stem cell microvesicles, ESM MVS, microvesicles. So I will call them ESMVs, which is shorter. So they have the GFP as well as the ESL, so we're looking at the RNA. And look at the protein on a Western, you can see the stem cells are the ones that have, the original stem cells have the GFP, and the microvesicles have GFP too. So they can carry whatever is in the stem cells, the microvesicles are carrying it. Now, since Mueller cells have been shown to have all the characteristics of the progenitor cells and can be transformed into different retinal neurons, including photoreceptors, we investigate if the transfer of the contents of the ESMVs to them could cause this transdifferentiation. So this is a culture of Mueller cells, that one, the one that says control. Is a culture of the Mueller cells. And we culture them, and we put the microvesicles on the cultures and started to look at what happened there. And then we saw the stem cell colonies, and we saw that cells were starting to have all these different shapes and changes from the plain Mueller cells. So there are morphological changes that occur. And we characterize those cells, and I will show you in a little bit that. But we were looking at the transfer of the contents of the microvesicles. So we look at the mRNA transfer, and 8 hours after the incubation of the ESMBs with the microvesicles, we saw, for instance, the
so
OCT4. Oh, good, thanks. We saw, for instance, there, that there is transfer. These are the Mueller cells that have not been incubated with the microvesicles. And these are the Mueller cells with the microvesicles that they have been separated. They have been washed several times to make sure that no microvesicles were there. So the microvesicles come in by, they are endocytosed practically. They fuse with the plasma membrane of the cells, and they deliver the content. And we have shown that already. It's all published material. So we saw that with OCT4 and with SOX2, and those are two pluripotent genes for pluripotency, as you heard just a few minutes ago. NANOG, for instance, another one, another gene for pluripotent, that is a pluripotency gene, is not, you can see, is not transferred. Practically, this is all the same thing. So if the transfer is selective, it doesn't mean that everything is transferred. There are things that are not transferred, and we have shown that too. Okay, so the mRNAs are transferred. Not only they are transferred, Mueller cells have very little, but they do have some OCT4. Mueller cells that we are using are human cultures of human Mueller cells. And we saw that there was an induction of expression of OCT4 using RT-PCR with primers for the human OCT4. And so the microvesicles are even inducing the endogenous OCT4 in these cells. They're also induce, they also transfer microRNAs from the stem cells. And you can see it here. This is the microRNA 292 and the 295 also. They are also microRNAs that are, they are pluripotency microRNAs and using pluripotency. They also induced the expression of early retinal genes in the Mueller cells, and you can see it here. PAX6 increases with time of incubation of Mueller cells with the ESMBs, and RAX, these are early genes in the microvesicles. It's going to take me another 5 minutes at least. I'm just telling you. Okay, so what we decided to do was to just do microarrays to see globally what were the mRNAs, and we did it also for microRNAs that were changing with the incubation of the Mueller cells with the microvesicles. And we found that there were pluripotency genes that were upregulated by these, OCT4, LIN28, KL4, LEAF. We found early retinal genes that were upregulated. You can read all those. We found notch pathway genes that activate progenitor phenotype in Mueller cells that were upregulated. We found retinal protective genes that were upregulated, inducers of retinal regeneration, and extracellular metrics modifying molecules that create permissive environment for tissue remodeling. They were all upregulated. On the other hand, we found genes that were downregulated, and these genes were the ones that promote differentiation and inhibitory ECM components, as well as inhibitory scar tissue components, and genes involved in driving retinal progenitors towards glial fate. So the microvesicles had a real, very good effect on the Mueller cells. So we corroborated all this by RT-PCR each one of those genes, well, the ones that I show you here, there are examples. Some were upregulated, others were downregulated, the same genes that I just showed you before. So we did the same thing with a microRNAs, and I don't need to spend the time on that, and some microRNAs were also downregulated. So we know that Mueller cells differentiate along retinal lineages we've showed also that they differentiate along retinal lineages after the incubation with ESMBs. And for instance, GAD67, which is a marker for amacrine cells, was very well expressed in these cells. And glutamine synthetase, which is a marker for the Mueller cells, we did the double labeling to show that the Mueller cells were the ones that really were were expressing GAD67, and this is the merge image, and you can see that there is here both GAD67 and G55. The same thing happens with NUN, that is a marker for ganglion cells, and you can see very nice colocalization of the glutamine synthase with the NUN mRNA. And then, for instance, BRN3A, which is another ganglion marker, was expressing very well in the Mueller cells. And you can see this is the control. The Mueller cells that didn't have, I didn't say that in the other two slides, the Mueller cells that were not incubated with the ESMBs didn't show any expression at all. The same thing happens with Syntaxin-1, another good marker of amacrine cells. And even rhodopsin, we could see some expression here, and nothing in the cells that were not treated with the ESMBs. So, and this now, I'm going to show this very fast, and this is nothing that is published. This is just what we have started to do. Can ESMBs augment the retina endogenous regenerative capacity? And I'll show you this very, very fast. So, because I have been already told that my time is over. So, this is in vivo. So we thought, okay, let's damage the retina so that we can see very well and very quickly if the ESMBs can do something. So we did the damage using NMDA. And NMDA is very well known. This is something that has been described many years ago, that damages the retina, especially the amacrine and the ganglion cells of the retina are completely damaged. So we then look at the function, and we look at the localization by immunohistochemistry. And so this is NMDA damage. You can see that the ones that are ESMB-treated retinas start showing BRDU, which is a proliferation marker in the ganglion cells. This is, we wanted to know what cell types in the NMDA damaged retina after ESMB-treated were proliferating. So we looked, now this is showing just the Mueller cells. CRA-LBP is a marker for Mueller cells. And you can see that this is when we look at BRDU, just to see the proliferation. And you can see here that the same cells are at 7 days after treatment, they are BRDU positive. And nothing, again, in the controls, absolutely nothing in the controls. And here, this is looking at Syntaxin-1, amacrine cells, marker for amacrine cells, and you can see the colocalization. Here, Syntaxin-1 with BRDU, nothing, again, in the control cells. And this is GAD67, and the same thing, you can see the colocalization. Here, the yellowish the look, and nothing in the control cells. So we wanted to know if there was any kind of effect. This is very recent thing, and we have done it in a few animals at this point. But look at this. This is the ERG of a normal mouse, not treated at all. And this is the recording from an NMDA treating. Look at the maximum This is the B wave, of course, and this is the non-treated eye damaged, and this is the treated eye damage. So this is a variation in the, but there is a tremendous difference with the normal. And when they are treated with the ESMBs, the blue is the treated. So look at 450 here, it was at barely 300, and here, well, this is the difference with the treated, both eyes of the same mouse. Okay, so we did 9 animals. In this worked. This is the average that we got, improved by 51%. Some of 5 were really incredible. I mean, we are so excited with this data, but 5 were very incredible, show about, 2 of them show about 70% recovery, and 3 of them, it was lower, so the average is about 51% recovery in the B wave, and this is damage in the B wave. So we are going to continue with this, and these are the people that work in this project. This is Alex Yuan, who did as a postdoc in the lab. This is Diana Katzman, who was a graduate student that already graduated, and this is Emma Stackpole, and a fantastic undergrad student that did a lot of work with us. That's it. Thank you.
Okay. So what I'm going to tell you about today are some of the cells that we've been working with for almost 10 years now, first from mice, and then from pigs, and now from human cells, retinal progenitor cells isolated from the neurosensory retina. So of course, there are lots of different approaches to try to treat advanced retinal degeneration. We're going to hear about all of them, I think, while we're here today. I'm going to tell you about cell replacement. So stem cell therapy is relatively new in the eye, but retinal transplantation has been here for many years. We had some discussions over dinner last night about some of the history of this field. Of course, it has a long history, not such a great one, in the sense that transplants have been performed in the past in human patients, and these transplants did not integrate with the host. So the problem with transplanting fetal tissue or fresh tissue is the lack of integration, the lack of connectivity between donor and host. Stem cells have immediately overcome that problem. They integrate very well, they make synapses, they make the kind of cells that you want them to be, but the key is controlling that differentiation. So differentiation is really the key to harnessing the plasticity of stem cells, and we don't have full control over that, and we're working towards that now. So some of the issues that come up when you're trying to develop a cell therapy strategy are listed here. They include safety. So we heard a lot about iPS cells. iPS cells, I think, are great for disease modeling. I think they have a long way to go before they're safe enough for transplantation in the clinical setting because of the presence of pluripotent cells contaminating the differentiation step. And that's something we don't have a control over at this stage. So any small subpopulation of pluripotent cells can potentially lead to tumor formation following transplantation, and we see that in our studies we've published in the eye using mouse-to-mouse studies. So not getting a tumor when you transplant a human cell into a mouse is not terribly surprising, but when you use a mouse-to-mouse paradigm or a pig-to-pig that has been done, then this small population of pluripotent cells can be a big problem, and that's the same for embryonic stem cells as well. So one of the problems that we had early on, we can isolate these cells from the retina, from the developing retina of, again, mice and pigs, and from human donors as well. How do we get enough cells to grow them into the populations that we need? We figured that out, and I'll show